All right, guys, welcome. This is Wes, your biology instructor. I'm going to take you through central dogma today, and it's a topic that hopefully you didn't have first semester, depending on if you were virtual in person. Maybe you did, maybe you didn't, but we're going to walk you through the process of going from DNA to proteins. So again, I'm going to work you through some things here. I'm going to draw some pictures for you. Hopefully it'll be pretty clear. And if you have any questions or misconceptions on Friday during the Q&A session, that would be the time to ask and clarify anything that you don't quite get. Okay, so today I'm going to take you through the process of central dogma of biology. Right, and this term dogma, it means like a central theme or idea or process. So again, this process is going to take the instructions of DNA and eventually take those instructions and make some proteins from it. So we're going to start by just drawing a cell here. Right, so no cell, no particular type of cell. We know that in that cell, you've got a nucleus, and then in that nucleus, you've got some DNA, right? And in that DNA are all the instructions on how to make proteins, right? And proteins are just little machines that do jobs within a cell. Okay, so central dogma is going to be going from these instructions, the DNA instructions in the nucleus, and then making those things into proteins. So just to provide a few labels here, we got DNA, and then we've got proteins. You can see there's like a mesh point here between these two arrows. We'll talk about what that means, but essentially we got two processes. We've got process one and process two. Process one is called transcription. We'll get to that. And then process two is called translation. We'll also talk about that. So the main idea here, what we're after is, again, we are taking instructions. So we're taking instructions in DNA, and we are going to decode those instructions, and we're going to make some proteins. Okay, so let's draw a simple T table here. Some things that we want to keep in mind as we work through this. I'm just going to take a stapler as my straight edge. And we want to compare, we're going to compare DNA and we're going to compare RNA because both are going to be heavily involved in this process. So DNA, right? You might remember that it's got two strands wound together to make a double helix, whereas RNA is just a single strand. The sugar in DNA, the D, stands for deoxyribose. Deoxyribose. And the R in RNA stands for just ribose. So we got a different sugar. When we talk about the nucleotides and the nitrogenous bases attached, DNA is going to contain the nitrogenous bases adenine, thymine, cytosine, guanine. And we just have those listed here, abbreviated. RNA is going to have adenine, uracil, cytosine, guanine. All right, so a difference there. DNA, there's really just one flavor. It's just DNA. But RNA, we have a bunch of different varieties, some of which are involved in this process and some of which aren't. So we have messenger RNA or mRNA. We have transfer or tRNA. We have ribosomal or rRNA. We have small interfering RNA. And then we have micro or miRNA. So the top three, M, T, and rRNAs, will be the ones that are involved in transcription and translation. The other small interfering and microRNAs are kind of beyond the scope of this particular course. All right. Next thing we're going to look at. So we're going to bring in a kind of a diagram or a clip right here. And we're going to look particularly at this piece here. So we're looking at this right here. This is a gene, and a gene represents a section of DNA that codes for a particular protein. Okay, so here's our DNA strand. This gene codes for any number of proteins. It could be 100, it could be 10, it could be 5. It varies, okay? So what we want to do is go ahead and define this. So that's what we'll do. We're going to say gene, G-E-N-E, gene. That's a section of DNA that codes for proteins. All right? And we saw that gene. And then what we're looking at is the process of taking that gene and then making a copy that we can take from the nucleus to the cytoplasm where a ribosome is going to find it. Right? The DNA is super important and really precious. If the DNA is compromised or insulted or damaged in any way, that could be an irreparable problem for a cell, depending on the extent of the damage. So the DNA is going to want to be kept in the nucleus. It's not going to want to be shuttled back and forth because there's lots of things in the nucleases out in the cytoplasm that could impact and insult the DNA. So we want to keep it housed securely in the nucleus. So we need to be able to make a copy that can be brought out of the nucleus to the cytoplasm and the ribosome where proteins are going to be manufactured. All right, so the process of doing that is called transcription. Transcription. And in transcription, we are going to rewrite. So we are rewriting the DNA code in a gene into an mRNA code. And it's critical to remember that this is happening in the nucleus because the DNA needs to stay in the nucleus. So what we're going to do here is we're going to sketch this out. I'm going to have my DNA here. So here's my DNA. I'll leave it kind of opened up there. Spot. This is my DNA. And then I'm going to have part of this that is going to be my gene. So let's say my gene begins here, or begins here and ends there. And what we'll see is the beginning portion of a gene is called the promoter, and the ending portion of the gene is called the terminator. We have the promoter and the terminator. And this, of course, is the gene. So let's say that there's a signal of some variety externally outside the cell that calls for a particular protein to be made. There's going to be a communication series of events that take place, and it's going to lead to something called transcription factors starting to assemble at the beginning, the promoter region of this gene. So we're going to label these things transcription factors. Transcription factors. And those things are going to start to accumulate around the promoter site of the gene. Those things will also serve to provide a bonding site for the item that does the heavy lifting here in terms of copying. And that's what this is going to represent. This is called RNA polymerase, Polymerase. RNA polymerase. It does the heavy lifting. It does the reading of the DNA and then the matching of the complementary RNA nucleotides. Okay, so this is going to work this direction. It's going to read the DNA and it's going to match the corresponding RNA nucleotides. And then out the back side is going to be my RNA that forms. So this is going to be the 5' and this is going to be the 3' end. And we only add new nucleotides to the 3' end. That's how we know that this is the 3' end. And it's going to continue like that. And again, what we'll do is we'll just put a little table here so we make sure we understand how this matching up is occurring. Right, so this is DNA, this is RNA. So anytime that RNA polymerase comes across a nucleotide with a cytosine, it's going to pair that up with a guanine. Anytime it comes across a guanine, it's pairing that up with a cytosine. Anytime it finds an adenine in DNA, this is the difference. It's matching it up with a uracil comes across thymine, adenine. So those are the base pairing rules that take place. Get that back on screen for you. Those are the base pairing rules that take place in this transcription process. Right. So let's pull back that animation and give that a look again. Hopefully this comes through for you in the video. If it doesn't, I'll link to it. Let's go like this. So again, here's our gene, right? Again, the whole idea is to take the instructions here in this gene, and we're going to manufacture a protein from it. We have a promoter region, we have a terminator region, beginning and the end of this gene. And again, the whole point of this is to assemble a protein. So we're going to go ahead and go past this piece here, and we're going to go back to where we see the gene. So again, there's going to be some signals, some transcription factors that can come in here and bind at the promoter site, and that's going to show the RNA polymerase where to bind in the initiation stage. So transcription factors, along with RNA polymerase, are going to bind at the promoter, and they're going to start opening up the DNA and reading the DNA nucleotides, and then bringing in the corresponding RNA nucleotides. And we can see that happening right here, right? So the RNA polymerase is opening up the DNA. It's looking at the nucleotides there, and then bringing in the appropriate RNA nucleotides in red here that you can see. So it's matching up. Any place it sees a C with a G, G with a C. Any place it sees an A, it's pairing a U, and T with an A. Right? We can see that base pairing right here in that frame. It's a good shot that's happening. So this is going to continue until it reaches the terminator. When it reaches the terminator region, this process will deconstruct and these materials be recycled. And this RNA will then continue to RNA processing, which we'll pick up tomorrow. So just give this a watch and a look, and then I'll have some more information, second piece of this for tomorrow. Try to break it up into manageable sizes, and then we can chat about it on Friday. So I hope that's helpful.